Okay, very good morning, my dear friends. So today's our eminent resource person, Dr.A.Kavitha Madam Garu, Assistant Professor, Department of Botany, RBBRR Women's College, Autonomous, Hyderabad. Madam is going to explain remaining topic. Madam is going to explain in Pteridophyta. So, Madam, please welcome and start your session and share your screen. Please welcome.
We'll complete with evolution of Sporophyte in Bryophytes. Hello, good morning to everyone. So this is Dr.A.Kavitha, Assistant Professor, Department of Botany, RBBRR Women's College. I would like to share today the topic of evolution of Sporophyte in Bryophytes. So which is also a very important topic for your competitive exam point of view. So just I'm sharing my screen. Sir, host disabled participant screen sharing.
Okay, ma'am. Okay. Nani, screen sharing you. Is it okay, madam?
No.
Okay, one second. Chinny.
It's here, Daddy.
Screen sharing. Yes, madam, please you share it.
Yeah, it's okay. Fine. So today we are looking into one of the important topic in Bryophytes. That is evolution of Sporophyte in Bryophytes. So I hope the screen is visible. Now what is Sporophyte or what is sporangium or sporogonium? Sporogonium here we are talking about this. Hello, am I audible?
Yes, audible, madam. Please proceed.
Right, 1 second. Hello.
Madam, audible. Please you proceed.
1 second, sir. 1 second.
Okay, madam.
Rajni Chapo. Sir, class. Yeah. No, for this. Right. So, what is this sporophyte? It is the next, the another generation in the life cycle of bryophytes, where we can see the formation of spores, and these spores on germination giving rise to the adult gametophytic tissue. Now, so this sporophyte we have discussed like in Markantia or Anthoceros. It is a structure which is going to consist of a foot region or a branch or an unbranched stalk or seta and a capsule region. Okay, so this is a capsule region which is enclosing the sporogenous tissue or the spore mother cells, what we call them as the sporogenous tissue, which are undergoing reduction division and producing the haploid spores. Now, so this is sporophyte is a very delicate and it is a short-lived and nutritionally dependent on its gametophyte. Okay, so where from this sporogonium is developed? Right from the venter, the archegonium venter of the gametophyte, within which the zygote will develop. The zygote, that is after fertilization of male and female gamete, the zygote is developed, the zygote is transformed into this is sporogonium or the sporophyte. So which we can say that it is a dependent, it is depending on the gametophyte for nutrition, and it will be a short-lived. Now, to explain about how the sporophyte is evolving from the primitive bryophytes to the advanced bryophytes, to explain that there are two contrasting views are there. One theory which is called as the evolution of the sporophyte through progressive sterilization of sporogenous tissue. This is called as theory of progressive evolution. And the another controversy view is say that a progressive reduction or sterilization. This we call it as theory of regressive evolution. So these are the two controversy views have been derived to explain the evolution of the sporophyte. Now we'll go with this theory of progressive evolution. So this theory we are also calling it as theory of sterilization. Now, what is the sterilization? I'm explaining that. So this theory, it was put forward by the scientist called as Bower in the year 1908 to 35, and it was supported by Cavers in 1910 and Campbell in the year 1940. So please note down these years and the name of the scientist. So these may be expected in your paper, in your exam. Every time 1 or 2 questions are asked from this evolution of sporophyte. Theory of sterilization or theory of progressive evolution. It was put forward by Bower, Cavers, and Campbell from the years 1908 to 1940. Now, so what is the main idea about this theory? What are the main theme or the objective of this theory? Is the sporophyte of the complex. So what is the main objective of this theory is the sporophyte of the complex forms like Funaria, Polytrichum, Sphagnum, they have been evolved due to the progressive sterilization of the fertile tissue of the simpler form. Now, what does it mean? So gradually, the sterile tissue will be developed. Okay, when so the complex, that is the sporophyte, which is having the capsule region, highly developed capsule in Funaria or Polytrichum, and a long seta or a bulbous food, which is embedded in the gametophytic tissue. All that sporophyte, it was evolved due to the progressive sterilization of the fertile tissue from the simpler form, like Riccia and the Markantia. So we look into this. So the increasing sterilization of the sporogenous tissue from simpler sporophyte of Riccia to the complex type of Funaria. So we all can. So this how the sterilization of this sporogenous tissue is happening. So this can be divided into the following stages. Like there are 6 to 7 stages in which we will discuss how the sterile tissue has been increasing right from the simpler forms to the complex forms of the Funaria. Now, so in the first stage, okay, so this complete evolution, this I can be divided into following stages. So in that, the first stage is, so the simplest known bryophyte, it was observed in Riccia. Okay, this is the primitive bryophyte, what we call as Riccia. Now, in the Riccia, you can see that the sporophyte is represented only with the help of the capsule. I repeat, in Riccia, that is in the primitive form of bryophyte, the sporophyte is being represented by only a capsule region. Okay, so there be no trace. So here we can see in Riccia, there is no traces of any foot or seta region. Now, when we go for the second stage, that is how the evolution is done. That is first in the first stage, in Riccia, we have seen that there is only the development of only the capsule. That is, sporophyte is represented only with the help of capsule region. Now, when we move on to the second stage, so this stage was seen in Corsinia, which is belonging to the Hepaticopsida order, where you can see a small sterile foot region is going to be developed. A small, not even the highly developed, a small sterile foot is going to develop. And the capsule is going to be surrounded by a single layer of jacket. But inside the capsule, some of the sporogenous cells, instead of forming the spores, they form sterile nutritive cells. That is, the complete sporogenous tissue is not developing into the spores. Only few cells of this sporogenous tissue, they will form the spores. The remaining complete tissue will be the nutritive in value. Whereas, a small, very small food development we see in the second stage of the evolution. Now, when we move on to the third stage of evolution, here the sterilization is seen in the Spirocarpus. So this is another genera where we can see the development of, that is the evolution, see how the evolution is carrying on. We can see the evolution of or the development of a bulbous foot region and a small narrow seta region, okay, which is in addition to this fertile capsule. This is a capsule region. If this capsule below that, you can see a small, very narrow seta and a bulbous foot. So this is the how the sterilization, that is the sterile tissue, is developed in the third stage. Now, when we move on to the fourth stage, so this stage is represented by Targonia. So here you can see the sporophyte is going to consist of a bulbous foot and a sterile narrow seta and a fertile capsule. And more sterilization of the sporogenous tissue is observed here. That means to say, majority of the sporogenous tissue is going to be developed into the spores as well as elaters. You can see here. There is a elaters you can observe. This is in the fourth stage. Now, fifth stage, this stage is illustrated by the Markantia. Already we have discussed this. How will be the Markantia sporophyte? So the sporophyte is going to have a bulbous foot, and this complete sporophyte is embedded within the gametophytic tissue, and it is going to have a seta and a single layer of jacket of capsule layer, and the sterile apical cap at the apex of the capsule. And we can see the complete sporogenous tissue is developing into the spores and the long elaters with the spiral thickening. So this is how you can see the development of you can observe this in the here. So this is a basic Riccia species where you can see only this is a capsule. Okay, and only see a mass of foot region. That's it. Now, when we move on to the next, that is the Spirocarpus. Here you can see a development of the bulbous foot region with a small narrow seta and the capsule region here surrounded by a single layer of jacket. And we can see the sterilization of the sporogenous tissue here. Some of these cells, not all the cells, but only some cells are developing into the spores, and remaining tissues is acting like a nutritive layer. Now, when we move on to here, Targonia, there we can see the complete sporogenous tissue is being converted, having a foot and the seta region. So this is the Markantia, where is a gradual development of the seta you can see here for conduction. And this is a long elongated capsule region. Many of the spores and elaters or spiral thickenings are going to be observed. Next is a similar like this in the Pellia also, where still gradual development of the seta and the foot region can be observed. Now, in the sixth stage, this is represented by the members of Jengar manuals of Pellia and Icardia. Here, more sterilization of the sporogenous tissue is observed, where the sporophyte is completely differentiated into foot, seta, and the capsule is going to have a multi-layered of jacket, and the sporogenous tissue produce a mass of elatospores and the diffused elaters. Next, in the seventh stage, you can see this is observed in the case of Anthoceros. So, in this Anthoceros, the reduction in the Sporophytas has been noted. That is a multi-layered capsule. The capsule will be multi-layered, differentiating into epidermis with stomata, and the chlorophyll cells. So we have seen in the anthoceros, that is sporophyte is in the form of a long column-like structure having a central columella. In anthoceros, the sporophytes, we call it as a hornwort due to the formation, highly developed sporophyte, which is a few centimeters long, emerging out of the gametophytic tissue. So we can see the reduction in the sporogenous tissue with a multi-layered capsule and an epidermis with stomata and the chlorophyll cells. Next, when we move on to the last to the final stage in the reduction of this sporogenous tissue, that is in the sterilization of the increasing in the sterile tissue. Here, this can be observed in Funaria and Polytrichum, where the highest degree of sterilization can be seen, where the sporophyte is differentiated into foot seta and the long seta and a capsule. And the capsule is having a sterile tissue of capsule consisting of, you can see so many sterile tissues here. Apophysis is there, operculum is there, many layered of jacket is there, columella, trabeculae are there, the wall of the spore sac, and even the peristomial teeth. So, when we observe in the rhizia, that is in the starting stage of the bryophyte, that is in the static genera, the primitive genera, the sterile tissue was only a limited. Okay, the entire capsule, that is, sporophyte is represented by a capsule and the sporogenous tissue developing into the spores. So, gradually, the sterilization is going to be increased with the increasing in the sterile tissue, like in anthoceros. When we go for anthoceros, in the anthoceros, you can see capsule will be multi-layered. Okay, and the last when we come to the Funaria and Polytrichum, that is in the 8th stage, more increasing of the sterile tissue has been noted. Okay, so this is the progressive evolution of the sterile tissue, or this theory is called as theory of sterilization. And so looking to this in the last stage, that is in the Polytrichum or the Funaria, the sporogenous tissue is only restricted to the spore sac. Whereas in the rhizia, the entire capsule region is the going to have the sporogenous tissue. I hope this you can be observed. So this is the anthoceros, you can see. Slowly the increasing in the sterile tissue can be observed. So this is the last, where this is in the Polytrichum or in the Funaria, you can see this is a Funaria, where the capsule, this is the long seta, foot is embedded inside the gametophyte. This is a capsule region having the multi-layered epidermal layer, and we can see the trabeculae, peristomial teeth, chlorophyllous tissue. Okay, so the restriction here we can see only the sporogenous tissue is in the only to the spore sac only, from which the spores are going to be developed. Now, so thus the sporophyte of rhizia is the simplest among the bryophyte with a very high proportion of fertile tissue and a very small sterile tissue. Whereas when we move on to the advanced forms or the complex forms of the bryophyte, that is in Funaria, Polytrichum or Pogonatum, we have seen a complexity in the sporophyte with a high degree of sterile tissue and decreasing in the fertility tissue. That is, the sporogenous tissue is restricted only to the spore sac in the advanced forms of the Bryophyta. Whereas in the simplest, only restricted or sterile tissue is only very, very little, but fertile tissue is more. So the evolution of sporophyte here in bryophyte, according to this theory, there is an progressive evolution of sterilization of the fertile tissue. That is, fertile tissue is going to be decreased as the complexity of the genera will happen. So this theory is referred to as progressive sterilization of the fertile tissue. I hope this is clear for you according to this theory. This is theory of sterilization or theory of progressive sterilization of the fertile tissue. Now we are moving on to the second contrasting idea or the second contrasting view regarding the development of sporophyte or the evolution of sporophyte. So this theory is referred to as theory of regressive evolution, or we are also calling it as theory of retrogressive theory of evolution. Now, this theory, it was given by the scientists called as Church, Kashyap, Goebel, and Evans. Now, so what is this theory? Regressive evolution. What does it mean? So this will be in opposite to the first contrasting view. So in the first contrasting view, what I told you, that in the primitive forms, there is the much amount of fertile tissue. And when we go for the higher advanced forms or the complex forms, this fertile tissue was decreasing. Or other way I can say, more of the sterile tissue was increasing. Okay. Fertile tissue, in the forms where fertile tissue is more, sterile tissue will be less. In the other forms, what I tell you, fertile tissue is less, sterile tissue will be more. Okay, so that was according to the regressive, that is the sterilization theory. Now, according to this theory, what they say that the simplest sporophyte of Riccia, it is considered to be as the most advanced type, which has been evolved by simplification or progressive reduction of the complex sporophyte. So regressive, they can say that it's an opposite. So when more fertile tissue and that will be the primitive form. Less fertile tissue and that I'm calling it as a complex form. But what this theory is saying, that simplicity of the sporophyte is considered to be as the advanced form. I told you that Riccia is a primitive. Funaria and Polytrichum are advanced or the complex. But depending upon the reduction in the sporophytic tissue or sporogenous tissue is considered to be as the primitive. అంటే ఇక్కడ చూడండి, రిసియాలో ఏమవుతుంది, రిసియాలో there is the most of the fertile tissue. Okay, there is much of sporogenous tissue is present in the Riccia, the primitive form. So as there is much of fertile tissue, these forms are considered to be as the advanced form. Okay, and this sporophyte, it was believed that this Riccia, the advanced type of sporophyte, it has been evolved by the progressive reduction in the complexity of the sporophyte. Ante Bryophyta, the complexity of the sporophyte is more. Okay, so as the complexity of the sporophyte is more, so from the complexity was reduced from back to front. Okay, higher forms of complexity is more. It was reduced when we come to the lower forms, that is in the Riccia, the complexity was reduced. So it was observed that Riccia, the advanced type of sporophyte, it was evolved from the higher forms where the complexity is more. So due to the reduction in the complexity, the sporophyte of Riccia was considered to be as the advanced type of sporophyte. Now here we'll see in the progressive reduction, that is the regressive theory. What does it say? The semi-parasitic foliose sporophyte gradually lost its leaves and they became embedded within the gametophyte. This is the primitive forms. So there is a gradual reduction of photosynthetic tissue in the sporophyte. Restriction or the absence of stomata only in the apophysis region. In sphagnum, we can see the stomata of apophysis, they are non-functional. The capsules of the higher forms like mosses, hornworts, it was observed to be of multi-layered, which is subsequently became single-layered in the Markantia. అంటే మనము కాంప్లెక్సిటీ నుంచి we came to the simplicity. మనం further in the first contrasting view, what we said, there was increasing in the sterility tissue and decreasing in the fertility tissue. This theory is based on, we are talking this about the reduction in the complexity, right from the advanced forms to the primitive forms. So more is the complexity, the primitive is the sporophyte. Less is the, that is very few sterility, much is the fertile tissue. So that was given by this theory of progressive reduction. Okay. So along with these changes, they have been observed that there is a gradual elimination of CETA and the food. Yes, we have seen that in Bryophyta, that is in the Funaria and Polytrichum, there's a well-developed foot and the CETA. As we move to the lowermost ones, that was reduced. Now, all these changes, they are accompanied by the increase in the fertility of the sporogenous cells. So this reduction or the elimination, it was accompanied by increasing in the fertility of the sporogenous cells. So this evidence from comparative morphology and genetics, they support that the simple sporophyte is Riccia. It is not an advanced, but it is a reduced structure. So starting Lemanaru, they said that Riccia is an advanced type of the sporophyte. But as compared to these changes, they have concluded that Riccia, it is not the advanced, but it is the reduced structure of the sporophyte right from the advanced forms. So thank you. So this is about the two theories of evolution of sporophyte in bryophytes. I hope this is clear. I'll just stop sharing. So any queries here? Any questions? Any questions from the participant side? Sure. Okay, fine. So I hope there are no questions from the participants. So shall I wind up? Hello? Am I audible? Participants, if you are unable to unmute yourself, please post your queries in the chat box. Okay, it's clear. Very good. Thank you. Right. Thank you very much for your kind patience listening. Tomorrow, I'll take up with the stellar evolution in pteridophytes. Thank you very much. See you tomorrow. So I'm winding up the session. Biotechnology, I don't know, some other madam or sir has been taking up these topics of biotechnology. Rajeshwari ma'am, please, you can post this in the group so that sir will answer your question. Okay, fine. I'm winding up my session. Thank you very much.
అయిపోయింది క్లాస్.